Hello everybody, welcome to the nervous tissue portion of our AMP1 class. This is a brand new organ system that we'll be focusing on, and while you will see somewhat familiar aspects of this organ system, by the end of today's lecture you'll see a little more detail, and the second lecture on the nervous system will truly take you to unexplored grounds. So first, let's say a few good words about the nervous system, whose function is dependent on the nervous tissue. So why is that when you look in your textbook, we have chapter after chapter from 12 to 17 all about nervous system? What could possibly make this so important? And this first paragraph here encapsulates a lot of that. So our nervous system is one of the main controlling organ systems in the body that assists with maintaining homeostasis in various ways. The other is the endocrine system that we will detail in AMP2, this is chapter 18. So how is it that the nervous system can maintain homeostasis? In a few minutes, we'll see a little finer grained look at the nervous system, and how to maintain homeostasis actually takes you back to chapter 1. We have looked at feedback loops. And the negative feedback loop is typically how our nervous system operates, with just like most every other feedback system in the body. So what does it take to maintain such a feedback loop? First, we need to detect the changes in the environment. You'll see that our nervous system has specialized cells to do just that. We have to, receptors were the first part of that feedback loop. The second portion of the feedback loop was a coordination integrating center. Our central nervous system plays that role in our nervous system. And then we had the effector organs actually carrying out the job of adjusting some kind of bodily function. And our nervous system is operating in that role as well. Some cells specialized for just like that sort of function. So our nervous system is, because of the ability to carry information, because of the presence of highly specialized cells that serve as receptors, as the integrating system, and the sending information to the effectors, our nervous system is eminently qualified to play a role maintaining homeostasis. And in addition to that, nervous system is responsible for our behaviors. This crosses over to the realm of psychology, and you all know that some behaviors can be modified by either therapy, which is psychotherapy, which is molding the nervous system through conversations or exercises, or pharmaceuticals that can also change behaviors of an individual. Our nervous system is the place where our memories are stored. As students, and I'm speaking as a former student, and I'm still learning quite a few things in other areas of life. So we could not possibly learn anything without memories, because without memories we could not recall our prior experience. So whether you're preparing for that world famous practical that's coming up very soon, or lecture exam 4, you do rely on this function of the nervous system. and how the nervous system is causing movement. You all have seen the neuromuscular junction in the muscle chapter. There should be no question about that. Our nervous system is bringing nerve impulses to animate skeletal muscles to make them contract. So a number of these typed out information of the slides are encapsulated in one illustration, so I'm going to go straight to that illustration. So here's this world famous illustration of the overall organization of a nervous system. A lot of information is encapsulated here. So let's start with that representation of that feedback loop to maintain homeostasis. So in this light pink box, there is a portion of the nervous system that works when we are conscious, and we are consciously aware and consciously controlling this aspect of our nervous system right here. This is called somatic nervous system. So where are the receptors? Well, take a look at this word in this blue box. We do have receptors. Those are called sensory neurons. Those are the specialized cells, and they detect a change in the environment. The information, what they are detecting, is passed on to the integrating portion of our nervous system. This is the central nervous system, brain and spinal cord. So this yellow box represents the integrating aspect. As we'll see a little later on, the neurons that make up these organs are called interneurons. So this is where decision making happens. And then our nervous system also has this motor portion where neurons carry nerve impulses. In the case of this somatic nervous system, to skeletal muscles. This is the only effector organs we consciously control of the body. So here is this feedback loop sequence. We do need very special action on the part of the neurons. We mentioned in the tissue chapter that neurons are unique, world famous for creating and conducting nerve impulses. And these arrows, whether blue or red, are all representing the flow of those nerve impulses. And we'll take a look at the portions of the neurons that enables them to create nerve impulses in the second lecture. So here is one aspect of maintaining homeostasis. If you want to put an example here, what are the sensory neurons that create a consciously detectable experience? How about vision? Specific sensory neurons in our eyes, in our retina, are giving us visual experience that we consciously experience. Let's say you are seeing that your dog is playing with their food bowl and spilling food all the way around. What are you going to do to maintain homeostasis, to interfere? Nerve impulses from your eyes will be passed on to the brain. In the occipital lobe, a decision is made that you got to do something about it. A different portion of the brain will be told to send nerve impulses to skeletal muscles, and you get out of your chair, go to your dog, and maybe quiet him or her down and straighten out the food bowl and pick up the pieces. So this is a reaction to a challenge, and so you are instructing skeletal muscles in this action to carry out a response to the change in your environment. You are consciously experiencing the change, and you are consciously directing the effectors, the skeletal muscles. So you have used your somatic nervous system. This little box, a little darker pink, is the autonomic nervous system. Take a look at these boxes and names. We have sensory receptors, sensory neurons. They send nerve impulses to the integrating center. And then we have motor neurons that send nerve impulses to the effectors. So what's the big difference here? Well, in the autonomic nervous system, none of the events are consciously detectable, and we have no conscious control over the effectors. So this portion of our nervous system operates out of mind, out of sight. This is an automated portion of our nervous system. Again, we are not consciously aware of the changes in the environment, and we cannot consciously control smooth muscle, cardiac muscle, and glands. So let's take a look at one example here. The autonomic sensory receptors are typically located inside visceral organs. What about regulation of the heart rate? So detection of heart rate and blood pressure is carried out by sensory receptors in the aorta and in those carotid arteries that you can feel along your neck. If you take a pulse along the side of your neck, you are feeling the pulsing of the carotid arteries. In those arteries, there are receptors that detect stretch, and if our heart rate or blood pressure is too high, those stretch receptors send nerve impulses to part of the brain. We'll be talking about this part of the brain stem, in chapter 14. And there the cardiovascular center, the organizing group or the integrating group of neurons, will send a nerve impulse to the heart through these motor neurons. And the instruction for the heart will be to slow down the heart rate. So here we send nerve impulses to cardiac muscle. And this feedback loop, this reaction to the change in the environment, was done totally without our conscious awareness or conscious intervention. So autonomic nervous system, same operating principle, but we have no conscious awareness of these actions in our nervous system. This third box here is even more mysterious. We talk a little bit about this in the digestive system in AMP2. In between some organs of our digestive system, such as stomach, small intestine, large intestine, there is a network of neurons that communicate, and this is all focused on improving the quality of the process of digestion. So this is the enteric nervous system. Again, we'll be mentioning this only in AMP2. So we'll be looking at the classification of neurons. Take a look at sensory, motor neurons. Again, interneurons are populating our central nervous system. So here is the main culprit of creating nerve impulses and conducting nerve impulses, the neuron. We will go into the functionally important part of the neurons, which will be molecules, ion channels, and pumps. In the second lecture, these cells are very large. Some neurons have extensions that go to several feet in length. And so the axons need support from the cytoskeleton, and neurofilaments and microtubules are tremendously important in maintaining nutrient flow, material flow inside these long extensions. When we talk about this artistic rendition of a neuron, here is the cell body. There are many short extensions. These are the dendrites. When you look at the direction of these black arrows, they are all pointing from the periphery to the cell body. This is how dendrites carry nerve impulses, electrical signals. So this is one directional movement. Will be very important again coming up in the second half of this in the second lecture. The axons represent yet another extension of the neuron that carries nerve impulses. The axons carry nerve impulses away from the body, and again you can see these dark arrows. So when the nerve impulse reaches the end of the axon, you can imagine a log-shaped skeletal muscle cell here, and here we are in the neighborhood of neuromuscular junction. So nerve impulses tend to be passed on to other cells, whether it's a muscle cell, whether it's another neuron, cells of various glands in the body, all that transmission of a nerve impulse will happen towards the end of the axon. You can see the axons are branching, and if you remember the illustration of the motor neuron way back from chapter 10, you notice that the single axon branched out to several skeletal muscle cells. All the skeletal muscle cells of a motor neuron are getting nerve impulses from a single axon. When we look at the shape of individual neurons, this is the shape that you see illustrated in your textbook lab manual most frequently. This multipolar neuron, we see this a lot inside our central nervous system, and a lot of the motor neurons leading from the brain or spinal cord shaped like this neuron. In the sensory portion of our body, we have some unusually shaped neurons. We have extremely short bipolar neurons. We had the most famous one that we explicitly will mention in AMP2 will be in the retina. We have a layer of these bipolar neurons. They are extremely short, and they're just passing on nerve impulse to the cell next to them. Much longer cells of the unipolar neurons. The length of these neurons, their axons can go on for several feet, and the cell body sits in a swollen part of spinal nerves next to the spinal cord. We'll come back to these neurons and the gross anatomical effect they create in chapter 13. So here is the, I hope now, somewhat familiar functional classification of neurons. The sensory neurons, they are detecting changes in the environment. These are truly the most important neurons. Without sensory neurons, we could not detect changes in our environment. Think about the neurons in your retina, in your inner ear. Without those, you wouldn't see and you wouldn't hear. So how important are they? They're unmatched in their ability to take a physical change in the environment like light or sound and turn them into nerve impulses. And there are a whole host of different sensory neurons. We'll talk about this in AMP2 a ton more. But you have seen examples of these. Meissner corpuscles, Pacini corpuscles are sensory neurons. In the case of the integumentary system, you saw these neurons take a physical stimulus such as touch, vibration, pressure, and turn them into nerve impulses. The interneurons are present in the central nervous system, brain, spinal cord. In our brain alone, we have 100 billion neurons. In our spinal cord, we have about 20 billion neurons, and most of those are interneurons. They participate in the integration function. And then we have, of course, the motor neurons. These are the neurons that take nerve impulses from the central nervous system and take it to the effector cells. You have seen the very end of motor neurons when we looked at neuromuscular junction, and the end of the neuron there was the end of a motor neuron, bringing nerve impulses to muscles. We also have to mention in much greater detail of the neuroglial cells. In the tissue chapter, we noticed that outside of this large neuron, there are hundreds of these dark, small specks. We agreed that they weren't dust, but they were cells, which we collectively called neuroglia. Here we are going to take a look at the 5 subtypes of the neuroglias, and we'll illustrate their importance. The central nervous system and the peripheral nervous system have different sets of these helper cells. One big difference, and here's one clinical significance, that neuroglial cells can divide. They can undergo mitosis. Way back in chapter 3, we mentioned that there are some cells that don't divide ever in the body. Skeletal muscle cells are typical examples of that. Neurons themselves tend not to divide. A very tiny percentage of neurons have the ability to divide at very restricted locations in our body. Typically, neurons just don't divide. Neuroglial cells, they are programmed to replace dead neuroglial cells, so they have the ability to divide. And for that reason, most brain cancers that you hear about, they're not always gliomas. This is one of the most frequent class of brain cancers, but most brain cancers originate from neuroglial cells that divide uncontrollably. So let's start out in the central nervous system, and the first neuroglial cell type is astrocyte. When you look at this image, you can see the astrocytes looks like little stars. That's what the astro supposed to mean. They have various functions. Let's start with this one, metabolize neurotransmitters. In a neuromuscular junction, we already used this term neurotransmitter. We named acetylcholine as the chemical that transmitted the nerve impulse from the synaptic end bulbs of a neuron to the muscle cell. So inside our central nervous system, there are dozens of chemicals that serve as neurotransmitters. So just like in a neuromuscular junction, neurotransmitters needs to be removed if we want to allow our nervous system to react to new stimulation or react to new changes in the environment. So the old, already released neurotransmitters need to be removed. And one way to remove them is to internalize them and degrade them inside astrocytes. So astrocytes kind of clear the field here, the space of surplus or unused and unwanted neurotransmitters, and they just degrade them. Regulating potassium balance. We'll see in the second part, in the second lecture on this chapter, that potassium ions are one of the ions, in addition to the sodium ions, that participate in forming an action potential nerve impulse. How much potassium is present in this space has a major influence on the ability to create a nerve impulse. So this is an important function, potassium levels, maintaining them normally. And astrocytes also provide structural support. The neurons themselves don't represent a sturdy material. So to give a little tissue integrity to brain and spinal cord especially, we need astrocytes to make connections and create a little sturdy matrix that is found in between the cells. And because astrocytes crisscross the central nervous system, they are also doing something totally unexpected. Astrocytes form a sheeting outside of blood vessels that come into the brain and spinal cord. Now don't think about the large blood vessels like the carotid artery from the side of your necks, pass through the temporal bone and enters into the cranial cavity. Those are gross anatomically visible blood vessels. Think about the blood vessels and take a look at the size of this blood vessel. It's just about the size of a red blood cell. So these are microscopic capillaries, and they have a sheet that prevents the passage of some materials from the blood into the central nervous system. This is a protective function. A lot of the times pathogens or their toxins are not allowed to enter the central nervous system, and we can say, hooray, our central nervous system is saved from many bad influences. On the other hand, the blood-brain barrier also prevents the movement of fatty acids into the central nervous system. Fatty acids is a commonly used nutrient for ATP production in many organs, including skeletal, cardiac muscles. Fatty acids cannot enter the nervous system, and so our central nervous system relies on glucose. We'll see some of the consequences of this a little later. And also antibiotics to treat a central nervous system infection. What about chemotherapeutic agents to treat brain cancers? Many of those are also not allowed to penetrate into central nervous system tissue. So those treatments, antibiotics, chemotherapeutic agents have very, very low or no effectiveness on the brain. Specially designed drugs are made in a way that they can at least somewhat cross the blood-brain barrier. So this is all coming from the astrocytes. Also inside our central nervous system are the genitorial cells, microglia. Way back in chapter 3, we looked at endocytosis and among others, phagocytosis. So phagocytosis is the endocytotic event when large clumps of debris from outside the cells are brought into these phagocytic cells, such as microglia. And microglia, with the help of their lysosomes, they degrade that debris inside the cell. So why do we need microglia? We don't have so many infections in our brain. We don't have so many trauma inside our brain that tissue damage would occur 24/7. Well, there's a little secret to our central nervous system, especially to our brain. We lose, through a natural attrition process, 100,000 neurons per day. So those neurons, those dead neurons, need to be cleaned up. Their leftovers need to be digested, and so this role keeps microglia busy. And so you're thinking that, I'm losing 100,000 neurons a day. What am I going to have left in 4 or 5 days. So let's do this little mental calculation. We're starting out with 100 billion neurons in the brain. We lose 100,000 neurons a day. So in a year, 365 days, we are losing 36 million neurons. If we live to be 100, that would mean that we would lose through this natural attrition 3.6 billion neurons out of 100. So even at this huge rate of cell loss, in 100 years we're losing about 3.5% of our neurons. So don't even try to send me an email before lecture exam 4 that, hey, Dr. K, I'm losing too many neurons. I can't really take this lecture exam 4. Now you know the math. You have plenty of neurons even when you're 100 years old, and I have to confess, I've never seen a 100-year-old student in my classes, so but even they would have the capacity. At any rate, so this is the role of microglia: constant debris cleanup. And of course, if there's an infection, if there is a concussion in the brain when neurons are die or other damage is occurring, of course they clean that up too. Well, this is something that I will show you a gross anatomical evidence for in Chapter 14. We will come back to these cells because we'll look at the gross anatomical clumping of these cells, ependymal cells. This is probably cells you never heard of. There is an interesting process going on in our central nervous system. A fluid, cerebrospinal fluid, is constantly produced, stored in cavities called ventricles in our brain, and this fluid circulates in a pattern that we explore in Chapter 14. So, okay, we said at the beginning that neuroglial cells have to support neurons in some way. So just how could cerebrospinal fluid support neurons in a brain and a spinal cord? Cerebrospinal fluid contains glucose, an energy carrier for our neurons. So that's one support. Cerebrospinal fluid also has an abundance of sodium, potassium, and calcium ions. So any ion deficiencies can be addressed by cerebrospinal fluid bathing our brain and spinal cord. So this is the role of the ependymal cells, and we again will come back to these cells in Chapter 14. The last slide in this lecture, I will show you the ventricles, at least the largest ones in the brain. So this is our first cell that is acting in the peripheral nervous system. So what is peripheral nervous system specifically? If brain and spinal cord are the central nervous system, then anything outside the brain and spinal cord are peripheral nervous system. Think about those Meissner corpuscles, Pacini corpuscles. Yes, they represent peripheral nervous system. What about those cranial nerves that we mentioned in Chapter 11? Cranial nerve 7, bringing nerve impulses to the muscles of facial expression. Cranial nerves 3, 4, and 6, bringing nerve impulses to the muscles that move the eyeball. Those cranial nerves are part of the peripheral nervous system. Now, in Chapter 15, we will talk about these clumps of neurons outside the brain and spinal cord. These clumps are called ganglia, and those ganglia need some cells to make them a little more sturdy, and satellite cells fulfill that role in the peripheral nervous system ganglia. The last two cell types have a shared role. This event covering the axons occur both in the central and in the peripheral nervous system. Now, in the central nervous system, it's the oligodendrocytes that do this. In the peripheral nervous system, we have Schwann cells. And you might be asking, why in a world would anyone give these two different names? You'll see a tremendous difference in the biology of Schwann cells with serious clinical consequences. So they deserve to be called by different names. So what is this axon covering to begin with? If you look at the axon, this yellowish rod, in most of our nervous system provide insulation for these axons. So these axons are carrying an electrical signal, and just like in your house, the electrical wires have this plastic insulation on them, preventing the jumping of the nerve impulse electrical signal from one axon to another. So in biology, we don't have plastic coverings. We have cells that take on the role of creating this covering. What is insulating is what's called a myelin sheet. So here's a cell, and this just happens to be a Schwann cell, and the cell wraps around this axon, and the cell membrane of this wrapped cell provides this insulation. There's a magic ingredient in the myelin sheet. So the Schwann cells and oligodendrocytes produce a lipoprotein called myelin. Myelin is allowing the cell membranes in several layers to remain stable. And so myelin is really important for forming this stable insulating structure. In the second lecture on this chapter, we will see that this insulation will have this unexpected effect of speeding up nerve impulse conduction through the axon. I did a screen capture of a video that I usually play in lectures. So here's the process of this myelination. Here's our blue axon, here's this brownish Schwann cell, and the Schwann cells grab onto the axon and start moving around it without letting go here. So the Schwann cells keep stretching and moves all around the axon, and here you see the Schwann cells have turned around this axon 2, almost 3 times, and now you can see the accumulating plasma membranes here with myelin protein in there. This is how this insulation is formed. Now imagine if an axon is 3-4 feet long, how many cells does it take to provide this insulation? But it is happening. Without insulation, the nerve impulse is proceeding slowly. It is noticeable in human beings in the first few years of our lives. Myelination, the covering of these axons, keep on proceeding. And one measure of it, if you have kids, you take them to the pediatrician and they look at their gross motor skills and fine motor skills. Well, the development of fine motor skills are the latest skill our kids obtain by myelination. Without insulation, fine motor skills are never reached, or they reach extremely slowly the level of perfection. So here's the artistic rendition in drawing of Schwann cells, and here you can see the movement of the Schwann cell around this axon. So one unique aspect of what Schwann cells do is when they form an insulating layer, this is extremely stable. This is called the neurilemma, this stable covering by Schwann cells. Now, why would I care if this is stable or not? Consider the following instance. Injury to the peripheral nervous system. Let's say somebody has lost a finger because of an industrial accident. A machine caught one finger of this individual. Finger is torn away. Of course, the axons that leading from the palm of the hand to that finger are all cut. And the axon in that finger will die. The Schwann cells form this stable neurolemma that maintain a living tunnel even in that finger that is torn off. So this is why it's so important with injuries of the peripheral nervous system, someone losing a toe, a finger, an arm. It's important to grab that limb and take it to the hospital, because that limb or finger can be reattached. And here is the kicker of the peripheral nervous system. From the palm of the hand, the stub of the axon can sprout into this reattached finger, and axon can regrow into this living tunnel we called neurolemma. So that finger can regain some mobility, that finger can regain sensation. So this is the action, the unique property of the Schwann cells, making the stable tunnel called neurolemma. Let me switch to the next slide. In the central nervous system, you see this plain statement, no neurolemma is formed. So let's take a scenario here. How about stroke? In stroke, a portion of the brain suffers oxygen deprivation and all the neurons die. Now, the neurons around that dead brain area has the option of growing into the dead area and rejuvenate connections, sprout connections. But because the neurolemma is not there, all the tunnels of the oligodendrocytes are collapsed and they are dying as well. So sprouting of axons into this injured area is not possible, and typically scar tissue replaces the part of the brain lost through a stroke. Imagine another injury to the central nervous system. Somebody severed a spinal cord. Here's a person dove into a shallow pool or fell off the roof and fell onto a side of the sidewalk, and the spinal cord is severed. The spinal cord severed ends are reattached to each other. Why isn't there regrowth from the brain or from the spinal cord side into the stub region of that severed spinal cord? That's because the neurolemma is not present in the spinal cord. Spinal cord is central nervous system, right? And no neurolemma is there. So any sprouting neurons are without guidance, and therefore it's an incredible difficult task to undergo rehabilitation and regain some function in the case of a severed spinal cord. So this is incredibly consequential. This seemingly little difference of forming a stable tunnel by the Schwann cells or an unstable tunnel of insulation by the oligodendrocytes. They both insulate. One is a little more sturdy than the other. So gross anatomically, this huge mass of insulated axons form a particular colored mass in our central nervous system. Here's our spinal cord, and the lighter colored areas in this artistic rendition are called white matter. White matter is bundles of billions of insulated axons, and that greasy lipoprotein myelin gives a light color to that area. Where there is no insulated axon, Now, what could that be? How about the cell bodies of the neurons? What about dendrites? They form a darker colored area. This is called gray matter. And many places in our spinal cord, it forms a butterfly or letter H shape. In the brain, the cell bodies and dendrites tend to be located at the edge of the cerebrum portion of our brain. That's the gray matter. But inside, there is huge bundles of insulated axons forming white matter. So I found a cadaver dissection image very similar to this. Let's take a look at some real stuff in addition to artistic rendition. So here is the frontal section of a human brain, and in chapter 14 we'll be calling these ridges gyrus, we'll be calling these valleys sulci, and you can see that the gray matter, this darker mass of cell bodies and dendrites, follow every nook and cranny on this cerebrum portion of the brain. When you go a little deeper, you can also see the streaming bundled axons forming white matter, much lighter color because of that insulation. This is also a good view. Take a look at the connection between the left and right hemispheres. It's obviously white matter. Huge bundle of insulated axons carry information for processing between the left and right hemispheres. The structure 11 here is called corpus callosum. We'll be seeing that on a number of images. And before we close this lecture, I do want to show you, here's number 10. Take a look at these cavities here. These are normal cavities called ventricles inside our brain, and ependymal cells that make cerebrospinal fluid are located inside these ventricles, and cerebrospinal fluid fills out these ventricles. So that's just a reminder of the special role of the ependymal cells here on this cadaver frontal section of the brain. So this is it for the first lecture on chapter 12. And if you have any questions, please don't hesitate to send me a message or email me.